Thanks for the introduction. So, just as a way of introducing myself a little bit more, I thought I'd just mention that Walter Eliza Hall is a medical research institute, first and foremost. So you might be wondering why I'm talking to you about computational type stuff there. Well, it turns out in modern medical research, computation is actually an extremely important part of analyzing data sets. And it's a particularly painful part because it's one that's been relatively recent in the evolution of research. So it's only now that they're starting to require all this sort of heavy computation and catching up with ways of modeling this sort of stuff. So, and one of the particularly painful things are these computational pipelines, which tend to be quite long and difficult in genomics research. So to start off with, let's try and define a little bit what bioinformatics is. This is how Wikipedia defines it. They basically say that it's a field that develops methods and software tools for analysis of biological data. But then they go on to say that that bioinformatics is also these pipelines, and basically defines bioinformatics in terms of these analytics pipelines that are repeatedly used, which is a bit sad because that's only a very small part of the whole field. But it also shows you how important these pipelines are to the actual progress in the area. So what am I explicitly interested in? Really, it's reproducible research. So then there are kind of two kinds of reproducibility. The experimental reproducibility, where if you do a different experiment, can you arrive at the same results? And then there's procedural reproducibility. Can I do the same thing on my data, run the same programs and whatnot, and arrive at the same answer? And that's what this talk's going to be focusing on explicitly. And in fact, even that has got two components, the sort of the computational environment and the execution part of it. Because you have to have the same environment and to do your execution in if you want to reproduce results. In this talk, I'll be talking purely about the execution part of it, though the computation environment is also an important thing that could, in principle, be integrated also into what I'm talking about. So what is a pipeline? It's basically a series of stages that convert data from one type into another type. So you have this sort of kind of flow of transformations and things that kind of pass down to what they call the pipeline. So here's an example from sequencing of the steps that you can do to processing. So first of all, when you do next generation sequencing, what you actually get is not an assembled genome. It's not your genome as such. What you get a collection of really short reads along the genome and multiple ones of them. So it comes off the machine as a collection of short reads. You then generally take these and you align it against a reference genome. In the case of human medical research, you would align it against a human genome. You would do a bunch of quality filtering to get rid of things that didn't map well or things that are duplicates and whatnot. Then you do something like covariance because you want to know, well, where is this person's genome different from the reference? That's what we're really interested in. But those calls are themselves not 100% reliable, so you may want to do further quality filtering on that. And then this whole sort of process is the actual pipeline that is used in this data analysis section. And what do we actually want from our pipelines? Well, they tend to be very long-running because each one of those steps is actually quite computation intensive. And so we really want to do these in parallel. We've got multiple samples. We want it to be re-enterable. So if our pipeline is broken at some point and computation stops at some point, let's say the cluster you're running on crashes and it needs to be rebooted, you want to jump back in at that point and continue onwards because you don't want to have to redo a day's work of computation to get back to that point and then keep going. You want to be able to jump into that point and keep going. And you also want it to be reusable. So you want your pipelines to be, or at least components of the pipelines to be reusable in different pipelines and on different samples across the data sets. And so the first thing, if you're coming from a software engineering sort of background, you might immediately think of build tools, right? Because Make, Shake, Scones, and Maven, that sort of stuff, they all give you this re-enterability. You can pause a build, you can jump back into it, you can change your inputs, and then it can pick up where it needs to and run your build for you, right? So why not use Make or something like that to do this? Well, and Shake. Shake is, if you haven't heard of Shake, it's an embedded DSL in Haskell that basically implements something a bit like Make, except with a lot of extra features as well. But let's take a let's start by taking a look at make. So here's a make file. You may have already seen one of these before, but basically the syntax here is that you have some output file before the colon, you have input files that are required to build it after the colon, and then you have the command that's executed to build that file. And you can have things depend on other things, so your final program, A out here, might depend on two other things which need to be built first, and those themselves depend on other things to get built first, which gives you kind of a dependency graph like this over here. So we have our final output executable, which depends on the object files, which they themselves depend on some source code, right? And this can be generalized because there's repetition here. These rules are the same, really. So you can use file name pattern matching and stuff with these percents to generalize these rules a little bit. But the thing to note here is that this graph, this dependency graph that's been built, is wider than it is longer. And the arrows are kind of flowing backwards along your dependency tree, right? But this is not the case in the real bioinformatics pipeline. So here's a real pipeline that I took out of a paper on identifying human neoantigens. And you don't have to read all these boxes, but it just gives you a general idea. Note that it is extremely long and not wide at all, right? It's mostly just one big stream down the middle here with a bit of branching off in places and data coming in around the corner here, but it's mostly just one long line. So this is already quite a different sort of scenario to what you have in the software build sort of tool area. The other things that to note here is that it's actually quite composable. So there are 3 or 4 big boxes here: prepare inputs, perform epitope prediction, integrate sequence, filter candidates. But inside those, there are smaller boxes which are also part of the pipeline. So we have this big outer pipeline that's being composed of smaller blocks in the middle. And the last thing to really notice about this is that each one of these concrete blocks, like alignment and variant calling, annotate variants, can be done with different tools. So you can swap out. So in this case, the authors are using BWA to do alignment, but that's only one alignment tool. You could do alignment with a raft of different other tools, and they give you different results, which can influence the outputs at the end of the pipeline, of course. Even worse than that, the parameters that you give these programs also influence what you get out at the end. So if you fiddle with the parameters a bit, you could end up with quite a different result coming out at the end. And so this all needs to be somehow tracked, right? And it's not, when I say that one program can do alignment and another program can also do alignment, they might be functionally equivalent, but they might not be equivalent in their input requirements, which is an important difference as well. Like generally, if you have a C compiler, or you have something written in C, you can swap your C compilers without too much of an issue because the C specification is kind of the same if we ignore language extensions and stuff like that. Whereas here it's not necessarily true. The input requirements might actually be quite different. So you want something that will catch any errors when you try and change to a different program if you haven't satisfied the input requirements. So just to summarize, they're long rather than wide. There's a wide variety of ways to complete a different stage, so you can swap out different tooling, but they also have different input requirements, potentially. There's a large number of parameters each one of these tools have. So usually there's a whole, like 10 parameters or so that you can set on these sort of programs, and each one of those can significantly change the outputs. They're very computationally intensive. So we're talking about days of runtime here on a cluster. And so it's very frustrating to wait 2 days and have your pipeline crash because you forgot to do something or because you misnamed something. So that's what we're going to try and solve here. And it's ill-suited to make because make and friends kind of relies on file name matching. And file name matching, well, as you saw from that big pipeline, we're actually not interested in most of the files in between. We're only really interested in the output at the end. We don't really want to have to come up with a consistent naming scheme for the entire pipeline. We'd like that to be handled for us automatically. And the other problem is that make and friends kind of do things backwards. They specify dependencies, not flow. But when you come to a big, long pipeline like that, it's actually much easier conceptually to think about flow instead of thinking about dependencies. You can think, I'm going to do alignment, then filtering, then calling, then more filtering, rather than thinking backwards in terms of its dependencies. All right, so Oh, there's another problem too, is metadata. So metadata is something that's very hard to handle with the file name-based build tool, because essentially you have to encode it into your file name somehow and then parse it out later. So this is kind of okay if your only metadata is the type of file is a JPEG. Well, you can look at the extension or something. But what if you have a whole bunch of different tags you need to track? So here's some of the examples that exist in the bioinformatics pipelines, whether something is sorted or not, what reference sequence you align against, because obviously if you align one sample against a human, another against a cat genome or something, you can't really combine them later on. That would be an error, because they're mapped against different things. Capture panels are another thing that come up, but also tool parameters, because we want our tool parameters to be captured in our build system as well. All right, so what is BioShake? BioShake is an embedded DSL in Haskell, built on top of Shake, which is itself an embedded DSL in Haskell. I'm not going to talk about what Shake actually is, but you can basically just think of it as make. For the purposes of this, it can be exactly the same. So you specify rules, which are like those colon rules, and then you have build actions that that satisfy those rules. BioShake has this very natural syntax, so this is a fairly literal sort of code here, align pipes, or align to sort to call. That's something that we want to be able to write literally into our code, because then it's very obvious what we're doing. And we're going to catch as much as we can at the type by moving everything into the type level, so that the compiler finds these errors for us, instead of us finding out in 2 days' time when our cluster dies. It also does a bit of abstraction so that you can run jobs locally on your current machine, but also submit it to a cluster, because you might want to test locally and then submit it to something with a bit more computational power. And it's file name independent specification. So if you look at this align sort call syntax, there's absolutely no mention whatsoever of the file name, because we're not really interested in naming most of these intermediate products. It's also actually quite a lightweight layer on top of Shake, which is pretty cool. All right, so here's the core data type. This is pretty much it. So the idea is that these arrows that this flow from A to B is important. So let's capture that directly with a data type. So here we have a data type to represent stage A being piped, the output of stage A being piped into stage B. Okay. So we're going to represent the arrow directly as a data type, and each stage is itself going to be a data type. And then there's two type classes, so we all know what type classes are, thankfully. One is which is something I'm going to call pathable, which means you can take something and map it to a file on disk name, so a path name, basically. Not the greatest name, pathable, but at least it's okay. And the second thing we need is a way to map things to build actions. So we need to be able to say if we're going to do this stage, then that's going to result in this action. So action is like executing a program or opening files, doing something, doing some sort of action that will create the output given the inputs. Okay. And this is the example pipeline that we're going to end up with. We're going to explicitly have named outputs. So instead of having to name everything, we only name things that we want to keep and that we're interested in. So here we're saying we're going to align sort dedup call and then save it as something called output. So how do we represent a sorting stage? So sorting is a relatively straightforward thing. It involves invoking a command. So let's see how we would wrap it up into this sort of type class framework. First, we declare a data type to represent our stage, source. It's just a data type, so it's got no parameters or anything. And then we define a pathable instance. So we say we're going to map A piped into source is going to have a name attached to it, some files attached to it, and we're going to let that be. We're going to pull out the inputs that we have. So that's the paths of A. That's the first stage, the stage preceding this stage. And we're going to hash them and just tack on the end sort.bed. Okay? So that's a way of mapping this stage to files in a way that's dependent on the inputs that you give it. This is just an example. Later on, that kind of gets done for you automatically. Second thing you need to do is tell it how to actually build it. So we say that this bit here is actually buildable. given that we can name our inputs and outputs, and that our input is a BAM file, so that's a type of file. So this sorting program only works on BAM file inputs, so we can specify that we must have a BAM file here for this to actually be buildable. Then you just get the inputs and you get the outputs, and then we just call this command SAM_tool_sort, pass it the inputs and save it to the outputs. And finally, there's this instance down the bottom here where you say that the output of a sort file of the sort stage is a BAM itself. Okay? Because sort takes a BAM, sorts it, and then creates another BAM. Now, a lot of that is fairly boilerplate, so you don't really want to write most of this because most of these rules are kind of fall into the same sort of pattern. So BioShake has this way has some template Haskell to make life a bit easier here. Instead of having to write all that stuff, you can just create a data type. You can write a function which takes a data type, some input files and output files, and then actually does the command, and then you invoke a couple of template Haskell things to generate all that stuff you saw in the last slide, basically. So this is saying that we'll make types for sort which map to a single output file, and it's these type classes are required on the sorry, that one shouldn't be there. This is required on its input. This is a tag that gets attached to its output. This is how we build it. And this empty list here is for transient tags, which in this case there are none. So they're tags that if they hold on the input, then they'll hold on the output. Okay? So we can capture those things too. What's nice about this is it's very easy to capture a lot of this metadata that I've been talking about. So file types is one of the very simple sort of metadata stuff that you can capture. But there are other properties too. So we can capture things like, has it been deduplicated? Is it from a set of paired end reads? Is it sorted? You can come up with any of these properties that you'd like to be able to capture and use them to enforce that they're satisfied whenever you need something, whenever a stage actually needs one of these predicates. There are other metadata. It's a little bit more complex. So let's say the referenced means that it's been referenced against some reference genome, and then there's some more stuff attached to it saying that, well, if you've referenced it against something, that something must have a location because it has to be accessed, and it has a short name as well, so like human or something. But what's really cool is that you can also very naturally capture transitive tags. I'm kind of abusing the word transitive a bit here, but you can say if we have something that's dedupt A, then anything that's downstream, A into B, is also dedupt. Because once you've deduplicated your data, you can't really add it back in, you've thrown it away, so it's gone. It's always deduplicated in the future. And same for paired end. If it's from a paired end read, then it's always going to be from a paired end read. There's no way you can not make it from a paired end read. And so this is a tag that can just kind of flow down the pipeline automatically. All right, so I thought I'd just give a quick demo of, well, we'll walk through what a pipeline actually looks like, and then hopefully it works and doesn't crash. So here's just a bunch of importing stuff. That's just usual sort of stuff. Here, BioShake has a small library for a bunch of tools, and BWA, Platypus, SAMTools, and Picard are some of those tools that's used in this pipeline. So we import them, give them some names. We define a data type to hold to refer to our input files, because you must somehow refer to your input files, and here we do that by explicitly creating a data type, which we're going to call sample, and the sample consists of 2 files in this case. Okay. The input files have some properties that hold on them. For example, they are FASTQ files, and they're from paired end. That's why there's two path names. So we can declare that the input files satisfy these predicates by making them instances of a type class. Same for referenced. We're going to reference them against something, so we just declare which reference we want to reference them against. And here we're just saying how to get our file names from our data type. We won't worry about threading because this is a simple demo, but BioShake also does handle a lot of multi-threaded stuff as well. And here's the actual pipeline here down the bottom. So hopefully you find this very readable. Basically, we take our list, our set of samples, and over each one of them we do alignment, mapping, sort, deduplicate, add some annotations, and then with all of our line files, then we do variant calling and save that result as calls.vcf out. So this is very, very readable. If you imagine a make file which did this process, it'd be very hard to see exactly which stages were being executed and what the actual flow was. But here it's very explicit. And we haven't had to mention any file names at all. We just mentioned in this last output one, and we haven't had to explicitly define any build rules or anything. So that's pretty neat. So what is this like when you run it on some files? So then I run it on some FastQ files, and we get that's the FastFQ files, the inputs, and we get this calls.vcf as the output. Okay. Now, obviously, the nice thing about this, as I said before, is that we really want to catch a lot of these errors up front. So let's say we forget to sort. Is it going to work? Well, no, because it's now a type error, right? Because you can't call unless you sort your variants first with platypus. It has to be a sorted input. So Haskell correctly infers that the sorted tag doesn't hold, and even tells you where the problem is. So it's a bit hard to read, but up here it tells you here that it couldn't deduce that the alignment is outputting a sorted file, right? So that catches it for us very nicely. Some of the other things that's very nice is that because of this abstraction where we're talking about stages now, not explicit tools, is that we can swap tools very easily. So instead of using dedup from SAM tools, we can call it from the Picard module instead, and then ask it to go, and it will call Picard now and generates our output file for us again. And it noticed, well, it might have been a bit quick for you to notice, but it didn't redo the whole pipeline. It continued on from where it could. So it continued on from the sorting stage. It didn't do alignment, map, sorting all over again. And the other thing that BioShake lets you capture is parameters. So like I said before, parameters for a particular stage can change your output quite drastically. So you want to capture those. Well, let's say we change our seed length in the alignment stage to 5 now, which is quite different from the default. Then it'll realize that, oops, forgot to save. It'll realize that now it has to go back to the alignment stage and realign, and then continue onwards to generate your output call. So these parameters are naturally captured. It also does some error. Unfortunately, not all errors are caught at compile time. Some errors are caught at runtime. So, for example, if I give it an invalid flag here and say that I want my seed length to be negative, which doesn't make any sense whatsoever, it will catch that error, but it will catch it. It's actually catching it at runtime, not at compile time. So this is good because at least it catches it up front before it actually executes the pipeline. But it's not ideal because you may have still had wasted time like copying this thing to a cluster and setting it up for execution, only to find that, whoa, hang on, I got a typo here. So ideally, it would be at compile time. All right, so just to conclude, BioShake is a pretty nice little DSL for modeling this sort of computational pipelines and stuff, but it doesn't capture everything. So one of the examples is what I just showed you. It can't capture this parameter incorrectness or parameter predicates and stuff like that. It even in the core sort of abstraction, I use just lists of file paths as which in itself is not great because they don't have a length in the type system, so you can't catch errors where you're feeding multiple files into a stage that's only expecting one file, for example. That can be solved if you move to like type length lists and stuff, but then you've raised the level of entry required to actually use this. So I wasn't sure I wanted to make that trade-off. The other problem obviously is it needs accurate metadata. If you incorrectly forget to say that your stage needs to have sorted data, well, it's not going to catch that error. So it kind of needs a well-curated library, if you want to call it that. Some things that could be done. Well, this abstraction is obviously not explicitly limited to Shake itself. It could be on any execution engine. So one of the ones that's gaining popularity in the field is something called Common Workflow Language. And you could easily take a description as written here and compile it to Common Workflow Language instead of using Shake as the execution engine. And finally, obviously, I want to have fully reproducible research here. So an integration with NICs and Singularity and stuff like that to capture the environmental reproducibility in addition to the executional reproducibility is kind of where I want to go. Just in conclusion, in acknowledgements, I'd like to thank my boss, Tony, for allowing me to actually do this work, even though it's not a major focus of the lab. And I'd like to thank Leon as well, who's in the audience. He's the other Haskeller at work, and we've had some great discussions. Thank you.
So we might have time for at least one question. Yes?
So you said that it supports clusters. I was just wondering if you could talk a little bit about how it distributes both embarrassingly parallel and non-embarrassingly parallel.
It doesn't do non-embarrassing parallel. It only does embarrassing parallel. So it's a coarse grain of parallelism, but it's something that suits this field quite a lot because usually you have a ton of input files you want to run the same pipeline over. And that you can just farm out to a cluster very easily. Each one of these tools also usually is multi-threaded as well, so that kind of gives you a bit of extra parallelism. And that is captured in the framework a little bit, so it does manage the maximum number of threads that you can have, so it doesn't launch like a billion jobs, which then themselves launch a billion threads and bring your machine to a halt. So there is a notion of finite resources.
How well does your model handle a process where a tool would generate 2 files and then you have to process them with different tools and then combine the results?
That doesn't fit into the arrow notation terribly well. You can handle it because you can explicitly define a stage that splits up files and then does different things on each one of them. So you remember I had this with all combinator which took everything in the example and combined it to one, which is then followed on. You could do the inverse. You could have a stage that splits things up and then calls 2 different pipelines in each one of those, and then potentially you could merge them back together later on. So it can be done, but it's not so natural in this arrow syntax.
I'm actually a clinician interested in reproducible research. Where do you think the burden of maintenance should lie? Because you mentioned that you're in execution environment dependent. Medical research sometimes gets quoted or repeated like decades later when things might be very different. And the people doing the research, doing it for their PhD might be long gone. Yeah. Should the tool makers, like the people of the BioShake, have some way of migrating their tool to use the newest execution environment, or at least validating that the input from the old environment is the same as the new one?
That's a great question. That's very hard to actually answer. I mean, at some level, it's already difficult even to reproduce your workflow in 1 year's time. Let's not pretend 10 years' time. So at the moment, we would go a long way even just using containerization like Singularity and stuff and the NICs sort of environment to actually bundle everything up so that it is literally executable again in 1 year's time. But ultimately, there does need to be a lot more effort put into this area.
All right. Thank you, Justin.